Hello everyone, this is Waad Nasr Al-Haqbani with my colleague Shahad Al-Ghilani, a Pharmacy interns from King Saud University, representing our poster with the title of The Potential Protective Effect of Alpha-Tocopherol and Lactobacillus Against Mercury Chloride Induces Hepatotoxicity in Rat. Role of CHOP, GPR87, and mTOR protein. First, introduction. Mercury is one of the most hazardous heavy metals in the environment. Most human exposure results from first consumption, dentin amalgam. Poisoning can result after a high exposure of mercury through inhalation or ingestion, leading to multi-organ toxicity, including hepatotoxicity. Although mercury is broadly sulfhydryl reactive, yet signaling cascade implicated in mediating in mercury-induced liver toxicity is not fully investigated. This initiates our interest to study a new mechanistic role of mercury hepatotoxicity at a molecular level and the way to its ameliorate using a combination of vitamin E and lactobacillus plantarum. For our objectives, we aim to investigate the hepatoprotective of vitamin E and or lactobacillus against mercury chloride toxicity by measuring the serum liver function test ALT and AST. Second, measuring the inflammatory biomarkers TNF-alpha, caspase-3, interleukin-6, and lipid peroxide GSH, SOD. And finally, the hepatic protein expressions of CHOP, mTOR, and GPR87 using Western blotting technique. For our methods, 25 adult male albino rats weighing in 150 to 200 grams were divided into 5 groups, 5 rats each group. The first group were the control that were only administered distilled water, while the second, third, and fourth and fifth group were all intoxicated with mercury chloride. However, the third group were treated with vitamin E, the fourth group were administered lactobacillus, and the fifth group with a combination of vitamin E and lactobacillus. Afterwards, blood samples were obtained and centrifuged to examine ALT and AST. Tissue samples also were homogenized and centrifuged to examine the protein expressions using Western blot and the ELISA and spectrometer to measure the inflammatory biomarkers of TNF-alpha, caspase-3. For our results from figure 1 to 4, mercury increases all the inflammatory biomarkers, while vitamin E and lactobacillus obtain the lower level. However, the combination has similar level to the control group. The same results are shown in figure 7 and 8. Moreover, in figure 5 and 6, the toxicity of mercury chloride reduces the level of GSH and SOD. On the other hand, vitamin E and lactobacillus alone prevent the reduction, while the combination showed a similar level of the control group. In Western blot membrane, you can see clearly how much mercury expresses the proteins, while in the treatment group, it prevented their upregulation, noticing that the combination showed a beautiful blend that can be exactly compared to the control group. In conclusion, vitamin E and lactobacillus provide protective effect against mercury chloride-induced hepatotoxicity by reduction of oxidative stress and inflammation, as well as downregulation of CHOP, GPR87, mTOR protein expression. Thank you.